Alright, so last week we pretty much finished up looking at cells and cell transport. So what I'm going to do today, I'm going to go through some past paper questions on cell transport and then move on to look at photosynthesis. So let's start with the questions here. So we have Mary and Suresh are playing with Mummy's perfume in the bedroom. Mummy smells the perfume as she is preparing dinner in the kitchen. So it says, name the process that allows the scent of the perfume to travel from the bedroom to the kitchen. Alright, so we agree that is diffusion. So remember, with diffusion, it happens in both liquids and gases. So this is a perfect example of diffusion in the air, which is the gas. So how does the process in A? So in other words, how is diffusion different from osmosis? Okay, so that's a good point, Shalom. So diffusion can happen without a membrane or with a membrane. So it doesn't need a membrane. So it can happen with or without. Any other points? So it's good to look at the definitions and compare. Well, it would happen through a liquid. Remember, I just said that the diffusion can happen in both liquids and gases. So it would occur through a liquid. Alright, so Shayla, you're saying osmosis is movement through water. But if you may need to reword that. There's something a little wrong there. So I think what you meant to say is that with osmosis, you're dealing with only water molecules. Diffusion, you're dealing with solutes, the movement of solutes. So that's the difference there. So just remember that osmosis is a type of diffusion that's only concerned with water molecules. So if we compare, so diffusion is a movement of solutes. Remember what solutes are? So solutes are the molecules that dissolve in water. So sugars, salts, anything like that. So diffusion is the movement of solutes, while osmosis is the movement of water molecules. So that's a difference. So diffusion happens in both liquids and gases. Obviously, osmosis will be happening in liquid, which is water, dealing with water. Alright, so those are some key differences. And with osmosis, you know that you require a membrane, a semi-permeable membrane. So diffusion doesn't need a membrane, but osmosis does. So those are two differences that you can mention. So then it goes down here to ask about active transport. So how would you define the term active transport? Okay, so the movement of molecules against concentration gradient, right, and requires energy. Okay, thank you, Myra. So that's the key, those are the two key things to remember. So the movement of molecules, so when you say against the concentration gradient, what does that mean? So let's explain that a little further. So we'll be talking about moving against the concentration gradient. Right, so from low to high, so I just want to make sure everyone knows what against the concentration gradient means. So that means the molecules, so normally just now we were looking at diffusion and osmosis. So typically molecules always move from where there's more to where there is less. So with diffusion and osmosis, you're going with the concentration gradient. You're going down the concentration gradient. Now with active transport, it's going against. So you're going from low to high instead of high to low. So low to high concentration. And then of course energy is required. So with the help of energy, and you can put in brackets the ATP, that's the molecule that carries energy and stores and carries energy. So that would be our definition for active transport. So the movement of molecules against the concentration gradient. So going from where there are fewer molecules to where there are more molecules. And you need energy to help pump them along. So usually this would occur across the cell membrane with the help of protein pumps. So those protein pumps that are found within the cell membrane will help to pump the molecules from low to high concentration. So state one part of the body where active transport occurs. So let's see if you have any ideas. Right, the nerves. So those are one of the key ones, key parts that you should know. Active transport occurs. Active transport can also occur within the small intestines, the muscle cells, but for sure the nerve cells. So we use that as an example. So anywhere where you're trying to get molecules that are in fewer amount to where there is more. So we have the nerve cells. What's the other name for the nerve cells? Right, so neurons, so N-E-U-R-N-S. So name the part of the cell where active transport takes place. So I just actually mentioned that, so you can answer the question. So which part of the cell would active transport take place? Alright, so the cell membrane. So generally speaking, in relation to all of these cell transport processes, diffusion, osmosis, active transport. As it relates to our cells, the cell membrane is what will be controlling what enters and leaves. So the molecules would naturally be passing across the cell membrane to get into the cell, get in or out of the cell. Right, so Myra, you just named some of the other areas of the body where active transport would occur, but for this one, they just wanted the part of the cell. So there's a difference. So one, they wanted part of the body, different parts of the body, and then they wanted the specific location within the cell that the active transport would occur. Alright, so let's move on to look at this question here. Alright, so part A is just listing the structures common to plant and animal cells. So what would be 3 structures common to plant and animal cells? Alright, so the cell membrane, for sure, all cells must have a cell membrane. It can't be a cell without a cell membrane. Cytoplasm. Alright, and vacuoles. But there's a difference in the type of vacuoles that would be in the plant and animal cell. So we put nucleus, so nucleus for sure. So in terms of the vacuole, what's the difference between the vacuoles and the animal and plant? Alright, so the other ones you can put, we have the mitochondria. Right, so the plants will have a larger one that takes up the majority of space inside of the cell. Right, so they're large, more permanent, and then within the animal is much smaller. But the truth is, yes, they both have vacuoles. It's just the difference in the size. Alright, so thank you, Regine, for adding that. Ribosomes as another one. So both of them will have ribosomes. What's the function of ribosomes? Okay, so it makes protein. So not so much that it holds protein. I don't know if I like that word hold. But so it produces proteins, made, synthesized. So any of those words would be better. So it's where proteins are made, manufactured. Alright, so let's quickly reviewing what's the function of mitochondria. Right, so that's the site where energy is made, where respiration occurs. The nucleus. Right, so where the genetic material is found, usually in the form of DNA or chromosomes. So DNA you will find it within the chromosomes. Okay, good. So redeem does give additional function there. So it actually controls all the activities of the cell. So we know the nucleus holds the genetic material, and is that genetic material that is responsible for determining what activities would occur in the cell. It would determine what type of cell it would be. So we looked at selected cells, nerve cell, epithelial cell, sperm, muscle cells. So each of those are specialized cells. So they would have a different set of genetic material that would determine the particular function of those cells. So yes, they would share similar DNA, but there are certain genes that will be turned on because of the specialized function for each of those cells. Alright, so let's look at part B. So let me zoom it in a little more. So we have a simplified illustration of osmosis. So we have this large container, so a beaker. We have a membrane separating two solutions. We have the water molecules and sugar molecules highlighted. So it says using one arrow, show on figure 5 the movement of molecules. So any volunteers? So you can use the whiteboard just to draw in the arrow. Or you can tell me from which side to which side, and I'll put in the arrow for you. That might be a little easier. So what direction would the molecules be moving? Right, so we have from A to B. Myra's saying from west to east. Alright, so we're going to have our arrow going like that. So that's one mark. So it says which part of the container A or B has the greater concentration of water molecules. Alright, so we agree that it is A. So you can clearly see that there are more water molecules in side A compared to side B. So side B is pretty much a sugar solution. So you notice that it has sugar molecules in sugar solution. So sugar molecules dissolved in water. So that's why you're going to see less water. So then the next part of the question says explain the process of osmosis using the illustration in figure 5. So it's important to use this information from the diagram to explain osmosis. I want to hear explanations from each of you. So I'm not going to say anything. So I just want you all to write it like if you were answering the question in the exam. So let's go. So full sentences and explanation, how you would answer it. So you have two answers. Dean, what's the problem? So let me hear from you. So you started saying you said dilute to concentrated. So I want a little more explanation on that. Remember, you have to use the illustration. So by use the illustration, they mean like within your answer, you should mention what's going on, what would be going on with the molecules in the diagram. Alright, so let me just comment on your answers. Alright, so all of you seem to have an idea of how you would answer it. And Myra, I know you went into a lot of detail, so that's good in terms of the passive movement. So you actually gave the definition and fully explain how the water molecules move, and then you said that the sugar molecules are bigger, so they won't be passing through the membrane. So all that is true. So what I'm going to do, so I'm going to type out the answer, my answer, and show you how we would go about answering it. So it says explain the process of osmosis using the illustration. So to make it as simple as possible, so you're basically going to merge the definition that you know and relate it to the diagram. So you will say a region of, or you can say from, if you're going to say from high to low concentration, you have to be specific to say water concentration, because that's where things can get a little confusing. Because usually when you think of concentration, you would tend to think of the sugar or salts, the actual solutes that will be dissolved in the water. In some cases you may see water potential, like a high to low water potential, meaning where there's more water to where there's less. So if you're answering it like that, you have to make sure you specify. So movement of water molecules from a high to low water concentration. From side A to side B. So you're linking up what you're seeing in the diagram. So you're going from a high water concentration, which is on side A, to a low water concentration, which is on side B, through a semi-permeable membrane. So you definitely can't exclude that part. So that's a simple way of explaining it. So you always have the water moving from where there's more to where there's less. So if you put it like that, that's fine. But it's just important to include the actual illustration. So I mentioned that it's going from side A, the water is going from side A to side B, and it's going through the semi-permeable membrane. Alright, so I will finish up there. This question, let me see if I can answer this one. This is connected to respiration as well, the respiratory system. But it says, name the process by which oxygen and carbon dioxide move across the alveoli. So let me see if anyone can determine which process it would be based on what we would have done so far. Okay, so it is gaseous exchange, but I think for this one they mean the actual process in terms of the movement of the molecules, which are oxygen and carbon dioxide. So you're correct, Myra. Gaseous exchange. Right, so diffusion is what they're looking for. So it's gaseous exchange through the process of diffusion. So remember, diffusion happens in both liquids and gases. So we're seeing how oxygen and carbon dioxide, they're both gases, and how they enter the lungs. The alveoli are special air sacs within the lungs. So we'll look at that in section B. Identify one way in which the osmosis differs from the process named. So we already looked through the differences between diffusion and osmosis. So that is it for the past paper questions. So hopefully everyone understands the differences between osmosis and diffusion, and also active transport. So you should know the definitions and know how to compare the three processes in terms of their similarities and their differences. So we know that osmosis and diffusion, they're both passive processes. They don't need energy. And you're going from high to low concentration. Well, active transport now requires energy, and you're going from low to high against the concentration gradient. Okay, so you said deals with air, so it can happen in air. So we looked at the example with the perfume, so that's an example of diffusion. So diffusion can happen in both liquids and air. So remember we give examples, so if you're making some Kool-Aid, you have the Kool-Aid powder or crystals or whatever, and you put them in water. That's an example of diffusion. You're going to spread out. So it's all about the solutes with diffusion and the gases. Well, with osmosis, you're dealing with water molecules only. So just remember that osmosis is a special type of diffusion. The only thing is that we're dealing with water molecules only, and it must happen across a semi-permeable membrane. The diffusion doesn't need a semi-permeable membrane, so it can happen freely or with a membrane. So in both cases, osmosis must occur with a membrane. Okay, so let's go on to look at photosynthesis. So who can tell me what photosynthesis is? So give me a definition. So from here on, we'll be looking at photosynthesis in plants, then go on to food chains and food webs. You see how plants are producers, how important they are. And then the final thing would be nutrient cycles. So hopefully we'll see if by next week we should be able to finish section A. Right, so how plants make their food. So that's as simple as you can put it. So how plants make their food. So plants make their own food. So what is required for plants to carry out photosynthesis? Alright, so Myra has all the details there. Okay, good. So we have, they need sunlight energy, water, and carbon dioxide. And Redeem also included chlorophyll, which is important. So remember, chlorophyll is the green pigment that gives plants its color. And that is the pigment that also absorbs sunlight energy. So we have the definition here. So the process in which plants use sunlight energy, water, and carbon dioxide to make their own food. So these are the raw materials. So when you hear the term raw materials, that's what they mean. So the raw materials are sunlight, water, and carbon dioxide. So those are the raw materials, and obviously chlorophyll is necessary in order to absorb the sunlight. So we see the sunlight is required, that penetrates the leaves where the chlorophyll is. We have carbon dioxide from the air that would enter the stomata of the leaves, and then the water from the soil enter the roots. So when we have all these raw materials, then the plant can use these raw materials to make glucose, which is the food. And then it also produces oxygen as a byproduct. So the main thing to produce is the glucose. But in the process of producing glucose, some oxygen is also given off. Excuse the annoying sounds of the dogs. So here is a breakdown of the process of photosynthesis. And the equation, which is important, you definitely need to know this equation. So we have the word equation and the chemical equation. So we're seeing here the raw materials are on one side of the equation. So carbon dioxide, 6 CO2, plus water, H2O, 6 H2O. Then we have the arrow, and light is usually, the sunlight is usually over the arrow, and you can also add in the chlorophyll as well, because those are other factors that are needed. So light and chlorophyll can go by the arrow, and then what is produced is the glucose, C6H12O6, and oxygen, 6O2. So we see how the raw materials will give rise to the product. So this diagram is just showing you how the water gets in. We mentioned that it goes through the roots, light from the sun, trapped by the chlorophyll, carbon dioxide would enter the stomata. So stomata are just tiny openings within the leaves. So that allows the gases to enter and exit. And then we have glucose being formed as the sugar that is formed. So glucose is a carbohydrate. So that usually is the product, but then you will notice that we have converted to starch. And this is important, especially when we go on to look at how you test to see if a plant is photosynthesizing. So usually starch is a good indicator that the plant has carried out photosynthesis. So we're going to look at the test for starch shortly. So we go from glucose, which is made by photosynthesis, and then the plant will pretty much convert it into starch, which is a storage molecule in plant. So this is just breaking down the two reactions that occur within the chloroplast. So remember, from the cell structure, you should know that chloroplast, that's where photosynthesis would occur. And you can see the chloroplast very well in the diagram here. So obviously they're green because they contain chlorophyll. So we have the two reactions that occur during photosynthesis. So the first one is the light reaction. So as the name implies, that means that sunlight is required for this stage to happen. So the sunlight energy usually will split the water molecules that it absorbs and produce hydrogen and oxygen. So this is the point where the oxygen gas will be released. And then the second reaction, you really don't need to know this in too much detail, but I'm just breaking it down so you see exactly what happens during photosynthesis. So then in the dark reaction, that means no light is necessary. So at night time, when there's no sun, the carbon dioxide from the air, when that gets into the plant, that is basically reduced by the hydrogen gas, and that is going to combine with the carbon dioxide to form the glucose. So it's just a breakdown to show you how each of the products are made. So once the photosynthesis has occurred, we know that glucose is produced, oxygen is produced as well. So what happens to the glucose after it's made? So that is what this slide here is showing you, the various things that the various ways that the glucose can be used by the plant. So we have to produce energy in respiration to build starch. So that's what builds up the starch grains in the plant cells. Then to build cellulose. So cellulose is that structural carbohydrate you find in the cell walls. So it gives the cell walls, helps the cell walls to maintain the shape of the plant cell. And then they can also be used to make amino acids. So amino acids are building blocks for proteins. So they're used for growth and repair. And then the lipids, also known as fats. You'll probably be more familiar with the word fats for sure. But lipids is the actual biological name for it. So that would be for storage and energy. So you're seeing how the glucose, so that main product of photosynthesis, how it can be used in these different ways to sustain the plant. And then the oxygen, which is the byproduct. So the main product is glucose, and we have some oxygen given off in the process. So the plant, because it's a living organism, it needs to carry out respiration. So it will use some of the glucose and the oxygen. The combination of those will help to produce energy. So we'll do respiration in section B. And then the oxygen that's released. So when you think of it, plants need us and we need plants. So the oxygen that the plants release, we need that oxygen. And then when we breathe out carbon dioxide, the plants use that carbon dioxide. So that's just a summary of what happens to the products of photosynthesis. Alright, so this is what I want to go on to now, the test in a leaf to start. It is a very important method that you need to know to test to see how a plant is photosynthesizing, if it is photosynthesizing. So usually you don't test for glucose. Although yes, the equation says carbon dioxide plus water give you glucose and oxygen. Remember I said earlier that the glucose is converted to starch. So in order to test to see if a leaf is carrying out photosynthesis, you don't test for glucose, but you test for starch. So what you're seeing here are the steps that you would carry out. And if you go into the course with the notes, you should have a video that actually outlines, that shows these steps. So you can watch that in your own time. I'm just going to break it down here. So the first thing you need to do, you're going to get you have a Bunsen burner, beaker, some water. You're going to need a boiling tube. So you get all your materials and your apparatus together. So the first thing you do is you set it up with the beaker of water on top of the Bunsen burner. So you're going to need to heat the leaf. So you have this boiling water, and then you're going to need to put the leaf, take a leaf from the plant and put it in the beaker of water. So what you're seeing here in this step, in this picture, is actually showing you step 2, but the leaf should be directly placed into the water. So that's what I was saying. When you get the chance, you can go and look up the video in the course. So the leaf, you put it in the water, the boiling water, and the whole purpose of that is that the heat is going to help break down the cell membranes, the cell walls. So we have to try to let iodine enter, and iodine is actually the reagent that we're using to test to see if starch is present. So you're going to need to get some of that. So before you can use the iodine, you need to take these steps first. So it's not that you just go and take your leaf from the plant and then start dropping the iodine drops on them. It's not going to work that way. If you were testing, maybe go on to do food tests, the same starch test in nutrition. So if you want to test, that potato has starch, rice. If you wanted to test food like that, you can actually just put the drops directly onto the potato. You don't need to heat it or do anything. But for the leaf, you have to heat it. Okay, so you did this at school already. Yeah, so this is definitely one of the main experiments we would do related to photosynthesis. So first step, place leaf in beaker of boiling water. So that's going to break down the cell membranes, allow the iodine to enter. And then you're going to turn off the flame and place the leaf in the boiling tube. So this was actually being shown here. So this could be a little misleading in terms of the picture. So this is actually step 2 here, where you're seeing the leaf inside of the boiling tube with the ethanol. So ethanol is an alcohol, and when that leaf is placed in the boiling tube, so the boiling tube is like a bigger test tube. So you place that leaf in the boiling tube of ethanol, and that boiling tube of ethanol is inside of the beaker, and the water will still be hot. So remember, it was boiling just now, but you turn off the flame because you don't need to have it boiling now. The leaf is already dead, so you kill the leaf, you break down the cell membranes and all that. So at this point now, it's all about extracting the chlorophyll, getting rid of the green pigment. Because if you just drop the iodine onto the leaf and it's green, that can offset the true observation as to what the color is that you should expect. So you won't be able to see the color change properly. So you need to get rid of the green pigment. So the green pigment, the chlorophyll, can mask the actual color that you'll be looking for when you add the iodine. So that's really the purpose of step 2. And then the third step, so once the color has been extracted, then you're going to place it onto a white tile. It could be a white tile or petri dish, whatever you have. So you put on your few drops of iodine, and if starch is present, you expect to see the iodine. The iodine is usually like a very brownish, yellowish color. So when you put the drops on the leaf, you should expect it to turn blue-black if the starch is present. So if no starch is present, you would have no color change. So that's what you're looking for when you're testing the leaf for starch. So you need to do these steps before you can actually use the iodine. So if starch is present, it should turn blue-black. So let's look at these. There's 2 investigations I want to look at, and then I will finish up. So the first investigation here, we're trying to see if light is necessary for photosynthesis. And obviously you should know that yes, sunlight is necessary because that's a factor required by the plant in order to carry out photosynthesis. But as usual, in biology you need to test things to see if these things are really true. So what you do, you take your healthy plant, you're leaving it in the cupboard for a few days to de-starch it. So this is pretty much to give us a clean slate. We're trying to get rid of all the starch that the plant would have stored up so that we start fresh, and we want to see if starch would have been created by photosynthesis while we're doing the experiment. So that's the whole purpose of putting it in the cupboard. When it's in the cupboard, it's not exposed to light, so it can't carry out photosynthesis. So therefore, the plant will start to feed off of the starch storage that it has. So that is what it means by de-starching the plant. So it gets rid of the starch. So if you're keeping the plant in the cupboard for those few days, it's not exposed to light. So it will eventually start to use up the starch storage. So once you do that, now you have a clean slate to start with. All the starch is gone. So then we're going to test the leaf for starch to make sure it doesn't have any starch. So if it doesn't have any starch, it should remain, when you put the iodine on it, it should remain the brownish color, no color change. So then the next step is fasten a folded piece of black paper or foil to both sides of a leaf on your plant. So you get the plant and then you choose one leaf. You can probably do it for 2 leaves if you want to. And you're going to put a strip of black paper or foil over the particular area of the leaf. You're going to expose the plant to normal conditions for a couple days. So normal conditions meaning it must get some sunlight, it must get some water, carbon dioxide, everything it needs to photosynthesize. So then after the few days that you gave the plant to photosynthesize, you're going to remove the cover from the leaf, and then this is when you will perform your actual test for starch. So you're going to test both areas, the part that was covered and the part that was uncovered. So the question says, what would you expect to happen? So based on all of that we just did, so we did our experiment. So when you come now to test the leaf for starch, so you're going to put iodine on the area that are exposed, and then iodine on the areas that have been covered up. So what would you expect to happen in this case? So I just want to see if you're making sense of this investigation, if you understand what the whole purpose of this investigation is. Alright, so Shalom has an answer there. So he expects the uncovered part to turn black. Okay, so the uncovered part you expect to turn, so you mean blue-black. So not just plain black, it's blue-black. And then the covered part would not react. So what do you mean by not react? You mean that it wouldn't have any color change. Alright, so Redeem is saying it creates starch. Okay, so the uncovered parts, the parts that are exposed, would have starch in them. Alright, so we seem to understand. So basically, the covered parts, you don't expect any starch to be there. So that area we would not expect to turn blue-black. Well, all the exposed parts should turn blue-black. So why would this be? So if you had to give a conclusion, so why? So we give the results, what you expect, the expected results. So why did the covered part test positive for starch? Sorry, why did the uncovered areas test positive for starch while the covered area did not? So let's think of what the aim of the investigation is. Okay, good. So the uncovered leaf, the uncovered areas of the leaf, they would have been exposed to the sunlight, along with the, you get in the water, yes, and the carbon dioxide, all that. So because they're exposed to light, they can carry out photosynthesis. Once photosynthesis occurs, they can produce glucose. Remember, glucose is the main product. And that glucose is then converted into starch for storage. So when you test that area of the leaf for starch, it is indicating that the plant has been carrying out photosynthesis. So you get that positive test. So these are the results of the experiment here, the investigation. So I already explained the purpose of the starch in the leaf. So after the investigation, you would expect that the parts of the leaf that are covered, it will not turn blue-black. No starch is present, so it can't turn blue-black if there's no starch present. And then the uncovered areas will test positive for starch. So this is answering our question that light is needed for photosynthesis. So if you cover up the entire plant, so like just as we did in the beginning, at the beginning we put the plant in a cupboard, it's not going to get any light at all. So even if you just did it and leave it at that, you can see that the plant actually would not be able to carry out photosynthesis because it doesn't have the light. So the fact that it's using up the starch storage, that means that it's unable to produce glucose, and no glucose produced, no starch. So that the starching part at the beginning helps to remove any starch stores. So this is what you would expect to see. So you see the covered areas looking yellowish color, and then the uncovered areas looking blue-black. Alright, so I will finish up there with that investigation.
If you found this video helpful, feel free to subscribe, like, and share. And don't forget to hit that notification bell.